This lesson's on absorption, and here are the objectives. Most of the absorption of nutrients occurs in the small intestine. And the small intestine has an enormous surface area increased by villi and microvilli. So villi and microvilli increase the surface area of the small intestine. Now what are villi and microvilli? Villi are projections of the mucosal layer of the intestinal wall. So if this is the intestinal wall, there are these finger-like projections that go into the lumen. And these increase the surface area so that there's more for absorption. So the lumen would be over here, and these finger-like projections called villi, singular is a villus, are projecting into that. Now if we zoom in on one of those villi, we see it here. So this is the epithelial layer of cells, and then inside we see a capillary bringing oxygenated blood, and then it's carrying away deoxygenated blood as well. And inside here we have what's called a lacteal. A lacteal is part of the lymphatic system being a parallel circulatory system, not carrying blood, but carrying fluid that is picked up from the interstitial tissues. And the lymphatic system also plays an important part in the immune system, which we'll talk about in a future lesson. So lacteals contain lymph, and there's a lacteal in the inside of a villus. So again, a villus is multiple cells, and it's this projecting outwards of the intestinal wall. And this is increasing the surface area. But if we zoom in on one of these epithelial cells as well, it itself has projections into the lumen, and these are called microvilli. So microvilli are projections from the individual cells, and then that is sort of superimposed on this villus here. So we've got villi like this, and then microvilli, and these microvilli are stabilized by the cytoskeleton in the cell. So we've got lots of ways to increase the surface area, and it's often quoted that the surface area of the entire GI tract is about the same size as the surface area of a tennis court. So lots and lots of area for absorption. Now if we think about the different kinds of nutrients that are being absorbed, they're going to different places. So the capillary is going to pick up the nutrients that are water soluble. So specifically it's going to pick up the sugars, saccharides, it's going to pick up the amino acids, and it's going to pick up peptides and carry those in the blood system. In contrast, the lacteal is going to pick up lipids. Lipids are fat soluble, not water soluble, and so they're not going to be able to enter and be carried away in the blood. So they're carried away in the lacteals, in the lymphatic system, and then that lymphatic system rejoins with the blood later. Now let's take a look at a few of the different ways that substances are absorbed. So again, I'm going to imagine that the gut lumen is up here, and this is the epithelial cell. So it's one of these cells here, and I'm looking at how the different substances are getting into that cell. So as you'll recall, some things can just diffuse straight through the cell membrane. That is, if they're fat soluble. So fatty acids, Fa is for fatty acids, and monoglycerides, which are one fatty acid connected to a glycerol, can diffuse straight through. So this is diffusion. Now, the fatty acids and the monoglycerides are formed by the breakdown of triglycerides, and then they cross the cell membrane by diffusion. Now, they actually get reassembled. They get reassembled into particles called chylomicrons. Chylomicrons are a packaging of these lipids together with some proteins, and then they get carried away in the lacteals. So, interestingly, the fats get broken down, triglycerides get broken down into fatty acids and monoglycerides, absorbed by diffusion, and then they get repackaged together into triglycerides, and together with some proteins get carried away in the lacteals in the lymphatic system. So fats are getting absorbed by diffusion. Now, some sugars, like fructose, can be absorbed by facilitated diffusion. So here's a facilitated diffusion carrier. This is facilitated diffusion. And so they're moving down their concentration gradient. Doesn't require an input of energy, but they can't pass straight through the membrane, so they need this facilitated diffusion carrier. In contrast, some other sugars, like glucose, for example, can't do that. Glucose moves into the epithelial cells of the intestine by a very interesting approach. Glucose is carried together with sodium, so both sodium and glucose go on the same transporter, and no ATP is being used here. However, on the other side of that cell, so imagine this is the other side of that cell, there's a sodium-potassium pump, and if you recall, the sodium-potassium pump pumps 3 sodium out for 2 potassium in, and it uses ATP to do that. So if you put the two sides of the cell together, what you see is that this sodium-potassium pump is lowering the concentration of sodium inside the cell, and as a result, this sodium wants to move into the cell, and therefore it can carry glucose with it, even if that glucose is moving against its concentration gradient. This is called secondary active transport. Secondary because the glucose sodium carrier here is not using ATP directly, but this sodium potassium pump on this side is. That's, again, lowering the sodium concentration inside, which means sodium wants to move down its concentration gradient, and that can be energetically coupled to moving glucose up its concentration gradient. So this is called secondary active transport, and that's how glucose gets in. Ions can be moved in by regular active transport using ATP, and water follows by osmosis. So again, these are kinds of membrane transport that we've seen before. And also, some peptides, some small peptides, can be brought into the cell by endocytosis as well. So these are small peptides. And some amino acids, the breakdown products of peptides, can also be brought in by this kind of secondary active transport mechanism that we saw here with glucose. So there's a lot of different varieties, a lot of different ways to bring things into the cell. So this just gives you a bit of an idea of the complexity at the cellular level of absorption. Lastly, I just want to return to anatomy to talk about the hepatic portal system. A portal system is a system where there are 2 capillary beds that are connected without going through the heart first. So here's one capillary bed, and although it's not drawn here, this is meant to be illustrating the capillaries that are coming from the villi in the intestine. So you can see here it says the capillaries of the digestive tract, and not just the intestine, but these other organs as well. So this blood here that's coming through the capillaries is picking up nutrients from the digestive tract. And it's carrying them not back to the heart like most blood goes, but stopping off at the liver first. So here we see this is called the hepatic portal vein. The hepatic portal vein is carrying blood with nutrients in it, and it's deoxygenated because it's also dropped off oxygen at the different digestive organs. So deoxygenated, nutrient-rich blood that it is carrying to the liver. And that allows the liver to process that blood and the nutrients in it before that blood is recirculated to the rest of the body. And that's important because the liver is involved in detoxification, and it's also involved in glucose metabolism. So having the blood stop off at the liver first allows these processes, as well as others, to take place before that blood is recirculated. So if we look then at the blood supply to the liver is getting blood from 2 different places. It's getting this deoxygenated but nutrient-rich blood from the gut via the hepatic portal vein, but the liver also needs a supply of oxygen itself, and it's getting that from the hepatic artery. So the hepatic artery comes off here from the systemic circulation and carries oxygenated blood to the liver to sustain it. Blood from both of these then returns to the heart via the hepatic vein. So the liver has this interesting blood supply of 2 inflows, the hepatic artery and the hepatic portal vein, and then 1 outflow, the hepatic vein. And you just need to be a little bit careful about the names because it can be easy at first to get confused between the hepatic portal vein, which carries blood from the gut to the liver, and the hepatic vein, which carries blood from the liver back towards the heart. So again, this setup is to allow the liver first crack at the blood that comes from the gut. So that's a look at absorption and the hepatic portal system.